This is video 7 for chapter 12, the nervous system. So in this video, we're going to look at the electrical properties of neural cells and start looking at how a neuron functions. So what I have here is a diagram of what the inside and outside of a neural cell is going to look like. So here is our plasma membrane of the neural cell. We know that it's a phospholipid bilayer, so two layers here, and water can freely go between these. But when a cell is at rest, and we were able to look at the ICF or intracellular fluid and ECF extracellular fluid, we would see that we have different concentrations of certain ions. So the inside of the neuron is high in potassium ions. So brackets around something means concentration. So we have a high concentration of K+. We also have a high number of negatively charged proteins because they can't go in and out of a cell because they're too big. The ECF is higher than the ICF in sodium ion concentration, calcium ion concentration, and chloride ion concentration. So because we have different amounts, we establish something called a concentration gradient. And all a concentration gradient is a difference in concentrations, in this case across a membrane, or from one area to another. Things tend to move automatically down their concentration gradients from high to low. But because we have a membrane, we might not be able to move things naturally through the process of diffusion from one area to the other. We would have to have something that would allow it to flow through this membrane. So we do have reasons for these concentration gradients. The first one is our sodium-potassium pump that we talked about in the first chapter. So we said that this pump was really important. It is embedded in the plasma membranes of cells. It's extremely important in a neuron because it is a form of active transport. It uses ATP and it moves sodium out of the cell and potassium in against the concentration gradient. Potassium normally wants to go out of the cell, but the pump pumps it back in to maintain that concentration gradient. But the membrane is permeable due to the presence of some channels. Remember that we could have channels that go through the membrane that allow certain things to go in and out. So the first type we have is called an ion leak channel. These channels are for the most part always open. Some of them have like a swinging door that allows things in and out. But they are very specific for one ion. So we have potassium ion leak channels. We have sodium ion leak channels. But we have a lot more potassium than sodium. That means that more potassium can move a lot easier than sodium can through ion leak channels. We also have gated ion channels. So these have a door that opens and closes in response to something opening or closing it. Well, we have different things that can react to open or close these gated channels. We have something called a ligand-gated channel. And we're going to see ligand again later on in the year when we talk about the endocrine system. A ligand-gated channel has a ligand, which is a molecule that binds to a receptor. So the receptor is usually extracellular, which means that it's on the outside of the cell. So it can be on the inside too, but for the most part, they are typically on the outside. So this molecule, this ligand, binds to the receptor. It opens the door. These exist for all 4 of the ions: potassium, sodium, calcium, and chlorine. So ligand binds to the receptor, gate opens. But we also have ones that work with voltage differences. So a voltage-gated cell means that we have to have a difference in charge potential, which is voltage, and it opens and closes in response to a change in voltage across the plasma membrane. So as we're going to see, there exists a certain voltage across the plasma membrane. There's a difference between the inside and outside of the cell. And if that changes, it can cause these gates to open. In an unstimulated neuron, so one that's at rest, the ICF is typically negative and the ECF is positive compared to one another. So if we had a movement of ions across the membrane, we could alter this resting potential, the difference in charge between these two, and the difference in charge, if it's big enough, opens or closes these gates and allows more ions to enter or stops them from entering. So we have all these different mechanisms that can open gates, but we do have a difference in concentration, which if we do open a gate, allows the ions to freely move from the higher concentration to the lower. So if we open a gate for potassium, potassium is going to move out of the cell. If we open a gate for sodium, sodium is going to move into the cell. If we open a gate for calcium, calcium is going to move into the cell because we have these concentration gradients up here established already. The sodium-potassium pump establishes the high inner ICF potassium and the high ECF sodium.